In interphase, we have discussed that interphase is completed in three phases, three further phases. For example, if we have a parent cell having 2n number of chromosome, mean having full number of chromosome. After interphase, when this cell passed from interphase, first step, it will grow in size. Second step, for example, if this parent cell have one ribosome and one mitochondria, for example, then after interphase, this cell have 4 in number of chromosome or 2 in number of chromosome having double stranded DNA, and the cell organelle is become doubled. For example, 2 ribosomes and 2 mitochondria. Right? After completion of interphase, the cell will go or enter in M phase. Now M phase will start. M phase mean mitotic phase. After mitotic phase, this one cell will be divided into two daughter cells, but each cell receive full number of chromosome. This process is called mitosis. So we can define mitosis as the type of cell division. If we talk about cell cycle types, cell cycle mean the division of cell into two or more than two cells. So cell cycle or cell division have two types. Cell division have two types, not phases, types. One is mitosis and the second one is meiosis. We will discuss this meiosis in detail, that it is composed of meiosis 1 and meiosis 2, M1 phase and M2 phase, and mitosis is just one phase which is called M phase, right after interphase. So this is mitosis, and mitosis, this is a type of cell division by which one diploid mean having full number of chromosome, mean 2 in number of chromosomes. One diploid cell divides into two. This is must now, because in cell cycle we say that the cell will divide into two or more. In mitosis, this is cell divides into two daughter cells, keeping number of chromosomes remain same. Right? Number of chromosome remains same mean that these daughter cell will be diploid, having full number of chromosome. Both have full number of chromosome. So for this reason, the cell will pass from interphase. It will become doubled, chromosome will become doubled, and then will split into two 46 chromosomes. Right? So this is mitosis definition. The second definition is a type of cell division in which one parent cell divided into two diploid daughter cells. It's called mitosis. The general overview of mitosis is this is, for example, we have this is parent cell. After M phase, the cell will pass from these four phases: prophase, metaphase, anaphase, and telophase. Right? After this parent cell will divide into two daughter cells. having full number of chromosome. After these four phases, this phase is called cytokinesis, in which the cytoplasm of the cell divides. Right? So these are separate phases. Mitosis is composed of or completes in four phases: the prophase, metaphase, anaphase, and telophase. We will discuss these phases in detail now, right? Because this cell already passed from G1 phase, S phase, and G2 phase. This cell is already passed from these three phases, and collectively these three phases is called interphase. We have discussed. Right now we will discuss. We have discussed the G1 S phase and G2 phase in detail. That it will take 8 hours, it will take 12 hours, it will take 3 hours or 4 hours, and this M phase will take 1 hour or 2 hours, right? So this is completed, and now we will discuss these phases. For this we will take a parent cell, and then we'll discuss that how these the changes occur in cells which is already passed from interphase, right? So remove comma. Mitosis mung nalagzed here nisisaka. Generally the cell is round, but for easiness we will take it in a square form, right? For example, we have a parent cell. This is parent cell, and this cell have nucleus, right? This is called cell membrane. Cell membrane. This is called nucleus, and we have discussed in structure and functions of cell that nucleus contains laminin proteins. Laminin proteins, for example, these are lamins proteins, and the cell already have euchromatins. Forget about prochromatin because they are not taking parts in cell division, right? So we will only show euchromatins, and these are euchromatins, and these are thread like DNA or chromosomes present inside nucleus, right? This area is called cytoplasm, and cytoplasm have cell organelles that we have discussed in cell. So it have cell organelles. Cell organelles mean ribosome, mitochondria, endoplasmic reticulum, Golgi bodies, lysosome, peroxisomes, right? And there is one cell organelle which is very important, which is called the microtubule development cell organelle, yeah, center, and this is called centriole. This is centriole. This centriole is connected by actin proteins with cell membrane, right? This is the connection of cytoskeleton. Cytoskeleton connects organelles to cell, and organelles to organelles, also cell to cell, right? So this is the connection by cytoskeleton, and this thing is responsible for formation of microtubules, and microtubules is actually spindle fibers, right? So this red boy here is called centriole, right? One thing other about this cell, that this cell is already passed from G1 S G2 phases, which is called interphase, and this cell is now prepared for cell division. Now it will start cell division. The first step in cell division is called prophase. But before we really go for cell division, there are some types of cells on the basis of cell division. Labile cell, proliferated cells, or permanent cells and stable cells, right? For example, we have labile cell. First, we have labile cell. Labile cells are also known as proliferated cells, right? And labile cells are continuously dividing, continuously dividing, right? What are the examples of labile cells? What are the examples of labile cell? For example, we have labile cells. Labile cells are epithelium of, the best example is epithelium of skin. Skin epithelium is continuously dividing because the cells present here are dying continuously. So the cells already dividing, right? For example, cells of GI tract, gastrointestinal system, GI tract cells are dividing continuously, right? For example, epithelium of urinary tract is also dividing continuously because the cells in urinary tract is facing acidic environment and they are dying continuously and therefore dividing continuously, right? Other cells, like for example, if we take stem cells of erythrocytes, RBCs, red blood cells, which forms myeloid cells continuously because red RBCs have no nucleus and they have specific lifespan of 120 days. After 120 days, the cells are degraded. So further cells are providing by bone marrows, and bone marrow make these RBCs from stem cells. So stem cells are also dividing continuously. For example, we have stem cells. Remember one point in your brain that all the cells are formed from stem cells. From stem cells, they are developing into white blood cells, into red blood cells, into platelets, into thrombocytes, for example, right? So these are labile cells which are continuously dividing, are continuously proliferating. These are also known as proliferating cells. The second thing on the basis of cell division is are stable cells. Second are stable cells. Stable cells cannot divide when it will provided by some stimulus or need, right? When it is provided by some stimulus, I have discussed the G0 phase. Also this is the resting phase. For example, if we have a cell here, right, and if there is no stimulus for this division, the cell is remaining in G0 or G0 phase because this cell is not dividing. It is not provided by some stimulus or some message, right? And there is no need of division. For example, if we have hepatocytes, stable cells are dividing when needed. It will divide when needed or when by some stimulus threshold, right? For example, if I examples of stable cells are, the best example is hepatic cells. Hepatic cells, also known as liver cells, right? For example, I have a full liver here, a full liver here. There is no need to divide these hepatocytes, but if I transplant some part of my liver to other, to recipient, for example, I have cut this liver like this, and I have transplant this part to other person. Now this is stimulus for division, and the cell will divide. So for example, these cells will divide now by mitosis, and the liver will completed. Here also this liver also completed. Right? So these stable cells divide when there is need. Right? These are called stable cells. The next type is permanent cell. Permanent cells. Permanent cells cannot divide once made. Right? These cells cannot divide lifetime. For example, the best example are neurons. Neurons. Second is cells of heart, myocardium, cardiac cells. Cardiac cells. Right? So these cells cannot divided lifetime once it's made. Right? These are called permanent cells. Right? Stable cell, labile cell. These are the types of cell on the basis of cell division, on the basis of mitosis. What is the importance of mitosis? Before we really goes to mitosis, we must know the importance of mitosis also. What is the importance? Wounds healing. Wounds healing. For example, if I got a cut here on my hand or foot, it will recover. This recovery is due to mitosis because the cells are dividing. Right? Bones formation, bones reformation. For example, I have a fracture bone, the bone is reformed. Right? For example, growth. Growth in humans, growth in animals, growth in plants is the function of mitosis. Right? This is the importance of mitosis. But if there is abnormal mitosis, abnormal cell division, it will cause cancer, because cancer, we can define cancer as the abnormal cell division. It's called cancer. So this is the clinical importance, that abnormal mitosis will cause cancer, tumors, etc. Right? Regeneration of liver after transplant. After transplant, when the liver is regenerated, this is also the example of importance of mitosis. Right? There are so many importance of mitosis and cell division. Right? So now we will discuss mitosis in detail. Before mitosis, we have learned the types of cells on the basis of cell division. Right? Run kam da. हाँ मखा भी बनाना हो यार. Now, this parent cell is already passed from interphase and now fully prepared to divide. So it will enter into prophase. For example, this cell enters into prophase. This is prophase, right? The parent cell, after interphase, it will goes into Prophase. Prophase, right? The first thing which take place in prophase is the condensation of this euchromatin, because this euchromatin is actually chromosome, and chromosome is composed of DNA plus histone proteins. Before division, these chromosome will become visible in cell, and this visibility is called condensation. The chromosome also grow in size, right? Because in parent cell, in germinal cell, these chromosomes are present in the form of threads. So it is called chromatins. These colorful threads, right? The first point is the condensation of chromosomes. Mean the chromosome become visible, right? The second thing, the enzymes which are produced in G1 phase, the enzymes which are produced in G1 phase will act on this nuclear membrane, because this nuclear membrane is formed of phospholipids and proteins, having lamine proteins, having nuclear pores, right? And now it will break down. So the proteins which are formed in, for example, we have proteases, we have proteases, we have lipases, we have amylases. These all are formed in G1 phase, because the protein synthesis take place in G1 phase. These enzymes will act on this cell membrane, this nuclear membrane, and this nuclear membrane become dissolved, or it will break down. So for example, this is our nuclear membrane. The second point is nuclear membrane break down and disappear. And disappear means dissolved. It is present in cytoplasm but not visible, right? The chromosomes condense, but now remember, in interphase, the amount of DNA become doubled, 2n. So all the chromosome must have 2n DNA, one centromere and 2n DNA. For example, if I have these four chromosomes, centromeres, right? And all have 2n DNA. In this case, two chromosomes are equal to 23. So 4 will be 46. Don't be confused, right? Because I cannot draw 46 chromosomes here. So these are 46 chromosome, and all chromosome have one original DNA and one copied. This is copied DNA, right? This cell have 2n number of chromosome, having amount of DNA is 2n, right? The next point, centriole is present here. This centriole divides and goes to opposite poles, right? But in some books you will find that this step take place in metaphase, right? In this phase only the centriole is divides, right? And spindle fibers appear, small spindle fibers appear. This is microfilament development centers. The centriole is also known as, microtubules tubules development centers, right? Abbreviated by MTDC, microtubules development centrioles. So these are divides and microtubules are formed, right? So this is the next step. The third step in prophase is centrioles mean microtubules development centers divides and moves, right? This is the completion of prophase. These three important steps takes place in prophase. After this we will go for metaphase, right? So this is, for example, the first step which occurs in metaphase is the formation of metaphase plate. So this is the metaphase plate. How metaphase plate is formed? Metaphase plate is formed by actin and myosin. For example, this red is myosin. This red is myosin, and actins are attached to this. It contracts like this. If I have a cell, this is, and actin and myosin contract. A cleavage is formed in the center of cell, and this cleavage line is called metaphase plate. So the first thing, this is metaphase. The first thing here is first metaphase plate formed. This is metaphase plate, right? The next thing, the centrioles goes to opposite sides of cell. Opposite sides are opposite poles, right? And the chromosomes are aligned on this metaphase plate. For example, we have 4 chromosomes. This is 1, 2, 3, 4. 4 chromosomes, right? These chromosomes aligned this metaphase plate, right? So the next thing is metaphase plate. When formed, the chromosomes aligned on metaphase plate, right? The next, the centrioles goes to, yeah, moves to opposite poles of cell, and the spindle fibers attached to chromosomes, right? How these spindle fibers attached to centromeres? There are some. For example, I have, this is centromere and this is chromosomes. This centromere have a hook like proteins like this, and these proteins are called kinetochore. These spindle fibers, for example, this is spindle fiber, it comes and attached to this kinetochore, right? So these spindle fibers are attached to chromosome. Here we have forgot one thing, 4 point, fourth, Spindle fibers formed are pairs. These are spindle fibers, right? And prophase. In metaphase, these spindle fibers were attached to the aligned chromosomes. This is a stepwise process. First, metaphase plate is formed. Second, all the chromosomes aligned on metaphase plate. The third, spindle fibers attached to aligned chromosomes. Right? Aligned mean in one line. Right? Now this metaphase plate is finished. The cell will enter into anaphase. Right? So for example, this is anaphase. But now this is the activity of actin and myosin. The actin gradually contracting and the cleavage is formed. So these are, for example, myosin and these are actin. They are contracting. Right? This is anaphase. There is one activity in anaphase, but really important. That is, for example, this is centriole. This centriole is attached by cytoskeleton to cell membrane, right? These spindle fibers will contract. These spindle fibers will contract and divides the centromere. For example, if I have divided this centromere, it will divide into two. So one full chromosome remains here and one full chromosome remains here. But now here the amount of DNA is one in here and one in here. This is the full story which is done in interphase. That in interphase the amount of DNA become doubled. In anaphase the amount of DNA will again become one in for separating the cell or providing the same DNA amount into data cells. So these are the centromere divides. First step, the centromere divides, right? And the chromosomes goes to opposite poles. For example, is the green part of chromosome and this is the red part of chromosome, right? I have discussed motor proteins in microtubules. Do you remember motor proteins? Anyone? Kinesin and dynein. This is actually the activity of kinesin and dynein. They are present here. For example, these are microtubules. Microtubules have two motor proteins. One is dynein and the second is kinesins. And these are called motor proteins because they are moving. Two types, dynein and kinesin. So it contract the spindle fibers in opposite directions. Opposite direction means that these proteins contract the spindle fibers in this direction and these contract the spindle fibers in this direction. Right? Now one active site will be on centromere. For example, this is dynein protein. Dynein will hold the centromere like this and will the second hand will be on centriole and they contract. When they contract, the centromere divides and goes to opposite poles. मतलब मेरा रे कना चिदा mata उस dynein kinesin will kana dagi karu moving kana. For example, we have two in DNA and these chromosomes. Now, and the number of chromosomes as 2n. Number of chromosomes is 2n are collectively it is called 4n for easiness. Right? Here, one end DNA moves here and one end DNA moves here. Right? This 4n, for example, we have 4n chromosomes. This is 2n actually because we have 46 centromere. But from 4n, 2n chromosome goes here and 2n chromosome goes here. Right? So each cell gets full number of chromosomes, but amount of DNA is also same as in parent cell. Right? Because before passing from interphase, this cell have 1n DNA. After interphase, this cell have 2n DNA. And now after full cell division, the daughter cells will get 1n DNA from 2n DNA, which is formed in interphase. Right? So this is anaphase. And anaphase, one point is important, that spindle fibers contracts and divide the centromere and moves to opposite sides of cell. Right? At contracted, centromere divides and moves to opposite poles of cell. Right? Now the cell will undergo telophase. So for example, this is telophase. Right? This contraction is made by myosin and actin. This is called cleavage furrow. This cleavage is called cleavage furrow. This is the site where cytoplasm well divides into two equal parts. Now, if you got MCQ in your paper that which phase is opposite to prophase, the telophase is solely opposite to prophase, because if, for example, this is telophase and telophase, here nuclear membrane break down and disappear. And telophase, nuclear membrane will reappear. It will reappear. So for example, we have, this is nuclear membrane reappear. Here spindle fibers appear. Here the spindle fiber will disappear. Spindle fibers will disappear. Spindle fibers are actually microtubule. So spindle fibers disappear. Right? Here the chromosomes condensed, mean visible, become visible. Here the chromosome decondensed. Chromosomes will decondensed. Right? So chromosomes will decondense. Right? So these are the point telophase. Now listen to me carefully. This cell have how many chromosomes in on board? Four. Four chromosomes. This cell have four chromosomes. This cell have four chromosomes. And have four chromosomes. This cell have four chromosomes. This cell have four chromosomes. So in definition, we have discussed that in mitosis, the daughter cell will get the same number of chromosomes as in parent cell, right? But in interphase, this is only the point which is very important, that in interphase this chromosome gets copy of DNA. Now if we take one chromosome from here and one chromosome from here, for example, this green chromosome is, this is the centromere and this is the centromere. This is the green chromosome and this is the red chromosome of this cell, right? And we take one chromosome from here also, from parent cell. For example, this is chromosome from parent cell. This cell have, this is original DNA, right? This is original DNA of this cell, and this is the copied DNA, right? Now after the full process, this centromere is divided. This is divided. One part of this centromere is here and one part is here, right? This parent chromosome have 2 in DNA. This daughter chromosome have 1 in DNA, but both the cell will get one original copy from parent cell. For example, in this the blue copy is, the blue strand is original. So both the cell have original strand of DNA and one copy is duplicated or replicated. So these both chromosome will get one strand copied DNA, right? This is the process by which the chromosomes divide, or the DNA divides into cells, and this is how the daughter cell gets full number from parent's cell, right? So this is telophase. After this, a process takes place which is called cytokinesis. This activity is continuous until the cytoplasm divides. Other thing here, that if parent cell have one ribosome and one mitochondria, after interphase this parent cell have two ribosomes and two mitochondria. After division, this cell will get one chromosome and this cell will get one chromosome. This daughter will get one mitochondria and this will get one mitochondria. Same like if after interphase this cell have, this parent cell have double number of cell organelles. So equal number of cell organelles which goes here and equal number of chromosomes will, cell organelles will go here. This activity is completed by actin and myosin fibers. So after cytokinesis, these cells separated, right? Nuclear membrane or nucleus. This cell have 2 in number of chromosome, right? These all cells have 2 in number of chromosome. So it will get 2 in number of chromosome. It will get 2 in number of chromosome. It have one centriole. It have also one centriole. It have full cell organelles and it will also have full cell organelles. Now this process is called cytokinesis. Cytokinesis means the division of cytoplasm, and these cells are called daughter cells having 2n number of chromosome, having amount of DNA is 1n. Right? This is the story of mitosis. Right? Any question? Huh? Anyone? But I have a question. If you don't have a question, I have a question. Why these are called sister chromatids? Why these are called sister chromatids, not brother chromatids? And why these cells are called daughter cells? Why not son cells?